our discussion of proteins. So I think this is where we stopped in class. Amino acids, definition, these are the monomers of proteins. They're carbon compounds that have both an amino group and a carboxyl functional group. So what I want you to do right now is to click pause on this video and sketch an amino acid, just the generic structure. Use the letter R for the variable group, but then get all the other pieces in place. And I want you to circle the two defining functional groups and label them both. So go ahead and do that now. Okay, presumably you have sketched an amino acid, and of course it looks exactly like this one here, where you've got your NH2 functional group on the left, you've got your COOH carboxyl functional group on the right. Your central carbon then has a hydrogen in the third position, and the fourth position is the variable R group down in red. Make sure you can recognize that, make sure you can reproduce that. So the next question we need to ask is, how do amino acids link together to form a protein, or a polypeptide, to be more specific? They're not considered a protein until they fold. So if you look up at the top, see if I can get a pointer here. If you look up at the top, we've got what we would call a dipeptide. So two amino acids. They are covalently linked through something called a peptide bond. So you can see amino group, central carbon, hydrogen, carboxyl group. Here's the variable R group for this one. It's got a CH2 called a methyl group and an OH called a hydroxyl group. The OH portion of the carboxyl group is now replaced with a covalent bond that we call the peptide bond. So the carboxyl group of one is covalently linked to the amino group of the next one. There's the amino group, central carbon, hydrogen, carboxyl group. This one hasn't reacted yet, so it still has its OH intact. And here's its variable R group. It's got a methylene group, and then it's got a ring structure, a benzene ring structure with a hydroxyl group. Bringing in a new or third amino acid, we're going to link it by dehydration synthesis. So we remove the OH from the carboxyl group, one of the two hydrogens from the amino group. There goes our water, our leaving group, and we're going to leave behind a carbon to nitrogen bond. That's what we see down here at the bottom. That's our peptide bond. Here's our amino group, central carbon, hydrogen, carboxyl group with its unreacted hydroxyl. And in this case, this is a cysteine, so it has a methylene group and a sulfhydryl SH group. So we build our polypeptides through dehydration synthesis between carboxyl groups and amino groups that create what's called the peptide backbone. You can see that name down here at the bottom, the peptide backbone. All these repeating units are identical. But what's waving out here in the breeze in each position is what makes each position unique. We call those the side chains or the variable R groups. Now, the other thing I want you to notice on this is that the growing chain has a certain direction to it. The very first amino acid put in place has an unreacted amino group. It will stay unreacted because all the new ones are going to react with the carboxyl group down here. So we've got the amino or N terminus. And then at the end, after the very last amino acid is put on, 200-300 amino acids later, we have an unreacted carboxyl group, fully intact with its OH associated with it. We call that the carboxyl or the C terminus of the molecule. Just like we see up top, all new amino acids are added by their amino group being attached through dehydration synthesis to the carboxyl group of the existing chain. Now, because there are 20 different possibilities at each position, if you had a short polypeptide of only 100 amino acids long, that would be 20 to the 100th power different possible combinations you could come up with, or 10 to the 130th power. That's a 1 with 130 zeros behind it. And that would be for a very small protein. So you can see that similar to what we talked about with nucleic acids, the potential diversity among proteins is practically limitless. Now, so far we just got a polypeptide. So far what we have is called the primary structure. Okay, the primary structure is simply the linear sequence of amino acids from the amino terminus up here in the corner all the way down. This one has 129 to the unreacted carboxy terminus. Okay, the N end and the C end of this chain. We call this sequence the primary structure of the amino acid, or of the polypeptide. Now, reality is that the R groups along here are going to want to interact with one another in different chemical reactions. And so if you got R groups way up here interacting with R groups way down here, for example, this cysteine number 6 can make a covalent bond with cysteine number 115, and that'll fold this together and covalently link it. All right, that folding that we see with all those interactions along the chain, we refer to as secondary and tertiary structure. If you take a more advanced course, then you'll learn the distinctions between secondary and tertiary structure. But for now, we're just going to lump the two together and talk about interactions among amino acids in a protein that leads to the folding. Now, some proteins only have through tertiary structure, while others have all the way through what we call quaternary structure. And quaternary structure is when two or more fully folded polypeptides have to come together in order to form a multi-subunit functional protein. So here's an example of hemoglobin. It carries oxygen and CO2 all around in our bodies. It requires multiple fully folded subunits before it has any final activity. So my suggestion to you now is to go back and replay this one more time. Take notes if you need to, pause it if you need to, look stuff up in your book if you need to, and challenge yourself yet again, quiz yourself on sketching an amino acid and quiz yourself on the different levels of protein structure.